Hello everyone, my name is Dr. Seung-Yong Lee. I am the course instructor for the molecular biology class, which is part of the essential requirements for the TSEP in biology program. I'm going to give you a little introduction about the course today. So this is a course syllabus. Here's my contact information, of course. So let's take a look at the course content a little bit. So this course is offered after students take biology class. So biology 105 is a prerequisite, and then the co-requisite for the course is chemistry 105. So at the same time, students will be taking chem 105 with this course. This is a 4-credit course, meaning the students will spend 3 hours of lecture every week, as well as 2 hours of lab every week. So the major course content, of course, will be dealing with majority of the biomolecules. So when I say biomolecules, what those are? Of course, those are the nucleic acid, DNA, RNA, as well as the protein molecules. And then there are different biomolecules like carbohydrates and lipids, but the main focus of the course is on the nucleic acid and protein because they play an essential role. DNA application, of course, how DNA molecules are made, and the transcription process enables making RNA molecules, and from there you make proteins through a process called translation. So we'll talk about the structure of these molecules as well as how those molecules are synthesized through those cellular processes. Of course, whenever those molecules are synthesized, there are strict regulation of those processes. So we'll learn about the regulation of those transcription and translation, as well as the mutation of the DNA molecules. So mutation is very important, especially in the field of biology, because mutation is the main source of the revolution, of course, right? And then we'll also talk about the differences between the prokaryotic cells and the eukaryotic cells. So when you talk about those central dogma, the main process is transcription and translation. Yeah, there are differences in how those processes take place in prokaryotic and eukaryotic cells. We'll also take a look at those. And finally, we'll talk about something about biotechnology, how the understanding of the modern molecular biology helped the development of the new industrial field called biotechnology. And then the textbook for the course, of course, is called Molecular Biology of the Gene, which is written by Watson et al. Currently, this is the 7th edition. This is one of the good books in the field of molecular biology, so I strongly recommend you buy a copy of it. So it doesn't matter, you can buy a used one or a brand new one. I know the textbooks are expensive, so you need to get one. And then in terms of how your course is graded, so I think that is the focus of your interest, right? So here, how your grade will be assigned. So throughout the course, there will be 3 lecture exams that will be given. So each exam is about 100 points. So 3 lecture exams combined will take up about 35% of your course grade, okay? And then you will have a lot of weekly assignments, pre-class assignment and post-class assignments. So pre-class assignment is probably the reading of your textbook material before your class time. So you have to write a summary, 1 or 2 page long. And then post-class assignment is at the end of the class. You will be given with a bunch of questions about the topics you learned. So you have to find the answers for those questions and bring those back the week after. So those are the pre and post-class assignments. They take up about 26% of your final grade. And then you will be asked to write a research paper. So the topic of the research paper is up to you, but if it has anything to do with the molecular biology, that is fine. So select a topic, and you need to collect about 20 of the research papers on the topic and read those, and you have to summarize what you read. So it's basically a literature review paper you need to write, and based on what you write on your paper, you will also give a presentation of the topic. So research paper and presentation will take up about 7% each for your final grade. So they are about 60 points each. And the course has a lab session with it. So through your lab, you will have 2 lab exams. So each exam is about 50 points. each. So combining 2, you will have 100 points. About 11% of your final grade will be coming out of your lab exam. And also you will write 2 lab reports. So those 2 lab reports will be out of so many labs you'll be doing. You will select 2 topics and write 2 scientific lab reports. Each report is about 30 points. So 2 reports will be about 60 points total. So about 7% of your final grade will be determined by your 2 lab reports you'll be writing. And then you will also need to keep a lab notebook throughout the semester. So I'm just emphasizing your experience about documentation, because documentation is really important. So through lab report and lab notebook writing, you will learn how to develop and maintain your documentation, so which is the evidence of your work in the lab. All right, so lab report, so every lab exercise you do, you have to maintain the record of it. So that's what it is in a notebook. So I'll be collecting your lab notebook about a couple of times throughout the semester. So you will be writing a bunch of lab reports, at least like 4 of them. So total 60 points will be given towards your lab notebook documentation. Okay. Finally, this is your course schedule. So as you can see, throughout the 16 week, you will be learning all these topics. So out of textbook, you'll be covering about 20 different chapters, okay, from chapter 1 through chapter 20. So each week you will be going through about 1 or 2 chapters, okay. So you will learn about a bunch of those biomolecules and their synthesis, okay, all those things. And the lab, of course, excluding the days when you have the exams, so you will go through about 12 different labs with different titles. So you'll be doing a lot of hands-on activities in the lab. So you will do solution making, aseptic cell growth curve observation, transformation of E. coli cells with PUC19 plasmid DNA, and the plasmid DNA isolation, and then DNA RNA protein quantification using spectrophotometry, and precision enzyme mapping of PBR322, and then you will do DNA methylation experiment, as well as DNA fingerprinting, and transformation of E. coli cells with plasmid, which will express the GFP gene. So GFP gene, when it's expressed, it'll make green fluorescent proteins. So after that, those transformed E. coli cells will grow in a liquid culture medium, and from those cells you'll be trying to isolate those GAP protein using a hydrophobic interaction chromatography. So you'll do a bunch of exciting hands-on labs throughout this course. I think that's it. This is a brief introduction to the course. I hope you will enjoy this class. I look forward to meeting you soon. Thank you.